So these platyhelminths are nothing but again they are primitive animals. So they have metazoan organization, and so here also the body wall of these animals have two layers, and the cells are organized into tissues, but there are no organ systems. Okay, like we have digestive system, respiratory system, or such kind of organ systems are not there. And animals of this phylum has three germ layers, like which called ectoderm, endoderm, and mesoderm. So even human beings will have these three germ layers. So during developmental process, so all the cells will come from these three germ layers only, which then organize into tissues, then into organs and organ systems, so on. So whenever a condition where we see these three germ layers, such condition is called as triploblastic condition. Okay, these are called triploblasts. Okay, and if not all organ systems are present, so like organs are present like tissues, but they are not like systematically arranged into any particular system here. And there in the platyhelminths, head-like structure appear for the first time. So in the other organisms which are more primitive than this platyhelminths, we don't see any head-like structure, a specific structure. Okay, and there is a mouth and a group of nerve cells which are called ganglia. So that means nervous system is slowly developing from this animal stage itself. So this arrangement is called cephalization. Okay, so but it is primitive one, not well developed, and they are also called as flatworms because they are dorsoventrally flattened, and they are bilateral symmetry. That means they can, if you cut them into half, you can see same kind of arrangement of the both ends. Okay, and they can be either terrestrial or aquatic. That means they can live on land or in water, and they are free living or parasitic. Some are free living and some are parasitic, means which live on some host. And all these animals are hermaphrodites, and there are acoelomates. There is no internal cavity. Okay, the space between various organisms is filled with the parenchymatous cells. Okay, again we will discuss all these terms and all these characters in the general characteristics. So platyhelminths are triploblastic, as I told, three germ layers, bilaterally symmetric, and acoelomates. Then they are flatworms, free living, parasitic, and they have hooks. Then chitin, which is made up of chitin. So these hooks and suckers will help them for their attachment. Okay, and the connective tissue is filled with the internal cavity is filled with the connective tissue which is called parenchyma. Then cilia or cuticle is present on the epidermis, so because for locomotion, and there is no special locomotory organ other than the cilia. Okay, and the nutrition is holozoic, so digestion is incomplete. Although it consists of mouth, pharynx, and intestine, but no anus. So the digestive system is incomplete. In some organisms like tapeworms, it is completely absent also. And respiratory and circulatory system is completely absent. Excretory system is present, but it is because of flame cells. There are specialized cells present in these organisms. These flame cells are called protonephridia. So you know, like in the kidney cells, nephridium, the term you might have heard. So here these flame cells are protonephridia, and excretory canal is present. And the nervous system is very primitive, although it consists of a pair of cerebral ganglia or a brain, but it is not well developed. So it has one to three longitudinal nerve cords. Okay, and in some organisms, even sense organs are present, and ciliary pits, ocellia, all these are the sensory organs for uh, detection of some senses. And most of them are hermaphrodites. And the most peculiar feature of this phylum are that presence of male and female reproductive organs. Okay. So fertilization is internal because in the same organism you can see male and female reproductive organs, and the fertilization is internal, and cross fertilization is seen in trematodes and self fertilization is seen in cystodes. And once it fertilization happens, it can form larval stages, and then this life this larval stage enters into a host and it completes its life cycle in one or more hosts. Okay, now we will look into the classification. So there are some controversies, like some scientists have suggested some classification and other scientists have suggested other classification. But right now we have three classes: Turbularia, Trematoda and Cystoda. So first we will discuss about Turbularia. So it's a free living. It occurs in moist soil, fresh water and sea, and they live in mud and ocean floor also. They are like leaf-like structure, bilateral symmetry. There are no segments here, and the body color is brown, gray, or black. Okay, and anterior end is called as head. It is differentiated. Epidermis is cellular, cells are present, and ciliated. Cilia is present. Like there are some rod-like structure which is called rhabdites. Then alimentary canal is present where you have mouth, pharynx and intestine. Okay. Then in some uh, like like except some turbularia, most of them are hermaphrodites and the development is direct with the juvenile form, and they are free living, and they emerge from egg or cocoon. So you know cocoon, like from caterpillar it forms a cocoon and from that it comes out. So like that here also some of them come from the cocoon and some of them come from the egg. Okay, and some are parasites and some are commensals. Examples are planaria and temnocephala. So here you can see how they look, the upper ones. Okay, turbularia. So first one A is planaria. So anterior you have anterior end like eye-like structures. Then you have posterior end is this end. Then you have tentacles, pharynx, and intestine and sucker. Then trematoda, next class. So in the trematodes, you have like adults are ecto or endoparasites, means which can live on other hosts, and they are elongated, and it has few forms of body are oval and some are round in shape, and they are unsegmented. There are no segments or scales. They have suckers and hooks for like for the attachment on the host. So they have two suckers, oral suckers near the mouth and ventral suckers near the posterior end at the bottom. And the epidermis is present and a thick cuticle is present like on insects. And the cilia and rhabdites are absent in the adults. Okay. And they have well developed digestive system with oral sucker. Intestine has divided into divided into two major branches, which again subdivides into various branches. And a primitive circulatory system is present. Here, excretory organs are protonephridia. The nervous system has a pair of cerebral ganglia and longitudinal nerve cords, and most of them has cross fertilization, not self fertilization, and reproductive system is well developed with two or more testes and a single ovary, and both testes and ovary are highly branched structures here. And in the ectoparasites, development is direct, while in the endoparasites, it is indirect. Okay. And the life history is complicated, having two or more hosts. Okay. Just a second, I'll continue. Sorry. So here the example is Fasciola hepatica. So this is the case study. Then we are going to discuss about this in detail. The next is cestoda. So cestoda are tapeworms. So even this also we are going to study. So they are flat ribbon-like forms. They are endoparasites. So endo means inside. So these parasites live inside the cells. Okay, that is cells of the host. So here usually in fish or mammals, so we can see these endoparasites. And these animals do not have any epidermis or rhabdites or cilia, but only cuticle is present. And three distinct divisions are seen in this tapeworm: scolex, neck, and strobila. And the body after the neck, so below the neck, whatever the body is called, that is called as strobila, and is divided into segments. They are called as proglottids. So the proglottids, which are present at the anterior end, that is at the top end, are small and narrow, whereas those at the posterior end are broad. Okay, and these proglottids can vary from around 4 proglottids to 4,000. And here we have segmentation, that is, you can see the divisions clearly, which are called as metamerism. Okay, so the basic rule of segmentation is the repetition of the same body parts in all the segments. Like if you see legs, some insects, they will have multiple legs. Okay, so they have like same legs are only present at the every segment. So same body part will be repeated at the every segment of the body. Okay, but segmentations, that is the usually basically the rule of segmentation is the same organs will get repeated. We can see that. But here, but segmentation is in cestodes is not like true segmentation. So that's why it's called pseudo metamerism. So pseudo means false. No evidence. So how we know that this is pseudo metamerism? Is evidence is that the cystodes, the new body segments are added in the anterior and anterior and new segments will be added. While in annelids, the new body segments are added at the posterior end. So it is quite reverse to that of the true segmentation rules. So that's why we call it as pseudo metamerism. Okay, and the anterior portion is differentiated into head, which is called as colex. But again here, it is not the true head. It looks like head because there are no sense organs. Okay, like we see like in our head, all the sense organs like eyes, ears, tongue, nose, everything is present in the head. Okay, and here mouth is present, but it's only anchoring organ, which helps to keep the parasite in the position in the tissues of the host. So it is for the attachment to keep up the parasite still at particular site of the host. And the oral sucker with hooks is present on the scolex, that is head. It helps in anchoring to the host tissues. And then a narrow neck is present immediately after the scolex. And below the scolex, we have proglottids, so which are immature. Okay, but at the middle ones are mature, and in the posterior end, they are called as gravid proglottids. Okay, these gravid proglottids, they will get shed off from the body during the process called apolysis. Like lysis means removal. Apolysis, like if you see in the upper, in the higher animals, apoptosis, like the cell death. So in the same way here, apolysis, like cell removal. Okay, and each proglottid is self-contained unit where it has male and female reproductive organs, nerve cord, and excretory canal with flame cells, which are excretory cells. And this supports the segmentation in the cystodes. So as I told you, it is again not true one. And digestive system is completely absent in the tapeworms. Excretory system is through flame cells, and canals are present. And nervous system is through a pair of ganglia in the form of a ring in the scolex, and two lateral longitudinal nerve cords are present, and all the cystodes excepting a single genus, which are like we will discuss what are the genus and all this. So they are hermaphrodites. So that means within the same you can see male and female reproductive organs. So a single set of reproductive organs are found in every proglottid, and large number of testes and a single ovary is present. And self-fertilization takes place. And here a sixth hooked stage, which is called as oncosphere, is present, is seen during the development. And the completion of life cycle can occur by having one or more intermediate course. So here example is Tinea solium. So this is tapeworm. So these two are very important topics. Okay, Fasciola hepatica and Tinea solium. So you can expect any question. So I'm not saying that this, whatever the important topics I am discussing, I'm not saying that they will definitely come in the exam, but they are important. You have to focus more on that. If you get time, you can also go through other topics. Because for small answers, you can expect from anything. But for essays, you can expect some important topics like this. Got it? So, first one is Fasciola hepatica. So this is the taxonomy or classification. You can see. So it belongs to phylum Platyhelminthus, class Trematoda, order Digenea, family Fasciolidae, genus Fasciola, and species Hepatica. So it is called as sheep liver fluke, and it is an endoparasite which lives inside the bile duct of the sheep and goat. So here, mostly in the areas where the rearing of the sheep and goats takes place, cattle, so there you can see this. So the history, this was observed a long back. So even in the 1883, a scientist called Thomas, he worked on this parasite. And the disease which is caused by this Fasciola hepatica is called as fascioliasis or liver rot. So liver rot because it causes a damage to the liver and bile duct of the sheep. Okay, not only in the sheep but horses, dogs, and other wild animals also. You can see this disease. Very rarely you can also observe this in the human liver because if the human they consume this contaminated food obtained from these animals, so they can expect like liver if they consume liver from these which is contaminated by this parasite, so even that can enter into the human liver also. So it has two hosts. The primary host is sheep, in which the adult flukes they attach to the walls of the bile duct, and the secondary host is the gastropod snail, which is Limnia truncatula, okay, where it completes its larval stage. So sometimes even snails, okay, Planorbis or Bulinus also can act as a secondary host. So now what are the external characters, how it looks? So it is soft, dorsoventrally it is like a leaf-like structure, dark brown color, which has bilateral symmetry. So most of these characters already we have discussed in the general characteristics. So I am not going to discuss in detail again. Just I will briefly go through. So the anterior end is broad and flattened. You can see in the image, and the posterior end is blunt. And it has in the anterior end you have a conical projection which is head-like lobe or which is called oral cone. It acts as a mouth, and the circular disc is present, which surrounds the mouth. So here you can see at one, which is labeled as one, and the two is oral sucker, a circular design. And then you have three, which is like head, and four, you can see gonophore, small hole-like structure, and fifth one is acetabulum. Means at the posterior end, at the bottom, you have excretory pore. Okay. So you have oral sucker, which will suck the food and for adhesion. And acetabulum is a ventral sucker, okay, cup-like structure. It is for the attachment to the bile duct of the host. And between these two suckers, there is a small hole which is called gonophore. This is genitalium for the reproduction. And the exterior, which is a excretory pore, that is mainly for the excretion of the waste. And the body wall, so body wall is, it is having a tegument, basement membrane, musculature, and mesenchyma. So tegument is nothing but it is a tough cytoplasmic syncytium. Syncytium is nothing but multiple cells each having the multiple nuclei. So if it is usually a single cell will have only one nuclei, but if it is having multiple nucleus, then it's called syncytium. And the entire body can withstand the action of digestive juices of the host, and it bears numerous minute and broad spines or spinules or scales. And it has a bile duct, and the, these small scales or spines they facilitate the locomotion. And beneath this tegument is a thin, delicate membrane, which is called basement membrane, and the musculature includes circular layer and a longitudinal layer. So inside longitudinal layer, there is another third layer which is called as oblique or diagonal muscle fiber, okay, which is made up of smooth muscle cells. Then you have the mesenchyma or parenchyma is also present in the internal organs, and no coelom. Coelom means the cavity, body cavity is not present here. Okay. Then irregular uninucleate and binucleate cells are present. And the digested food and waste substances are transported through this parenchyma, And as there is no blood is not present, so it cannot enter the circulatory system. So here only the parenchyma is present. Then you can see this is the digestive system. You have mouth, oral sucker, then you have muscular pharynx, esophagus, gonophore, acetabulum, all this labeled, okay. Intestinal branches, there are branches, and then ceca or diverticula and excretory pore. So your mouth is situated at the anterior end, that is at the top end, and then you have oral sucker. So this leads to a pharynx, which is a small narrow lumen with a thick wall, and radial muscles are present, and pharyngeal glands are present. And it opens into esophagus, which is again lined by a single layer of epithelial cells, and this opens into the intestine. Now the intestine is divided into right and left main branches or leaves. Okay, so on the posterior end you have the anus. Okay, before that you have ceca, okay, which is the outer side, which is a large branch structure, and then you have the alimentary canal, but there is no particular anus. So alimentary canal at the bottom end only there is an excretory pore. These luke forms feed on the bile fluid and blood, lymph and other tissue pieces which comes out of the tissues of the host. So these are all taken up by the oral sucker from the walls of the bile passages, and food which is sucked that goes into the intestinal ceca, and then the pumping action of, because of pumping action of this pharynx. So pharynx have muscles. So these muscles will have pumping action. And the digestion is extracellular. There are no cells involved here. And there are no complex enzymes, digestive enzymes present. But here, once the digestion takes place, it goes to the parenchyma because it is transported to parenchyma, but no blood is present. Then the waste is removed from the posterior end and the nutrients are absorbed. Okay. And the reserve food is stored in the form of in the mesenchyma and other muscles in the form of glycogen and fats. So it's a simple way of feeding and digestion. Then the respiratory system. So the respiratory system is anaerobic. So it doesn't require any oxygen. Even in the very little oxygen conditions also, it can survive. And the glycogen and glucose are the chief fuels. So energy fuels for the respiration. So whatever the glycogen which is stored in the digestion process, their metabolism actually gives the energy here in this process. So this helps in the movement and activities. So as it shows a very little movements, so it requires less energy also. Then next, the excretory system. So excretory system consists of large number of flame cells or protonephridia, which are connected to form the excretory ducts. And the excretion is mainly through osmoregulation. So what are these flame cells in excretory ducts? So flame cells are modified mesenchymal cells. So they are distributed very widely. There are many number of flame cells. They have elastic walls, branched cytoplasmic processes, and it contains single nucleus on one side and encloses a large cavity, empty side on the other end. And it has cilia. So when the cilia flickers, it looks like flame. That's why it's called flame cell. And the ciliary movement, they will excrete these liquid waste which are present. And excretory ducts. So the cavity of these flame cells are continuous with the capillary ducts that open into the tubules. Such tubules open into the excretory tubes. Okay, tubules to tubes. Okay. Then this anterior part of the body has four excretory tubes, which are two dorsal and two ventral, and they unite to behind acetabulum. So acetabulum structure we have seen already in the image I have shown you. I'll show you again. This. So the fifth one, okay, this is the acetabulum. And it forms an excretory canal. So it opens up through the excretory pore, and so whatever the waste material is present, that will be excreted. Liquid waste will be excreted out of this excretory pore. Okay. Then nervous system. So the nervous system is well developed here. Okay. But sense organs are absent. So they consist of collar or cerebral ring surrounding the esophagus, and the ring has a pair of cerebral ganglia and a ventral ganglia. And here the nerves appear, and which are supplied to anterior and posterior regions. And there are suckers. Suckers actually they receive more number of nerves. And three pairs of longitudinal nerve cords are extended posteriorly, and they are again branched here. And one pair is dorsal, one pair is ventral, and one pair is lateral. So here you have three pairs totally, nerve cords. So these nerve cords actually in the higher animals, they develop into nervous system. Okay. And they form commissures, that is branches, and they are usually bipolar. So this is the nervous system. So here you can see the one which is labeled as anterior nerves, and then two is cerebral ganglia, then three is brain, the ring-like structure, and then dorsal nerve cord. All are like branches only, okay? Fifth one is ventral nerve cord, then sixth one is lateral nerve cord, and seventh one is peripheral branches of the nerve cord. So this is the nervous system. So it looks primitive, but it is well developed, okay? Then reproductive system. So they are hermaphrodites, but can also cross fertilize. And the reproductive organs are well developed, and male and female genital ducts open into the common chamber, which is called as genital atrium. So it is situated in the anterior part, that is the top part of the body, and opens out through the common genital aperture or gonopore, or I have shown you in the image. And the male reproductive organs, excuse me, it includes testis, vasa differentia, seminal vesicle, ejaculatory duct, and penis. So testes are a pair of highly ramified tubular structures which are located one behind the other in the posterior middle part of the body. And the walls of these testes consists of germinal epithelium which produce spermatozoa like sperms. Okay. Then a duct which is called vas deferens comes out of the middle of each testis. And the two ducts run anteriorly and fuse at the level of acetabulum and forms a seminal vesicle where all the sperms will be stored. And a narrow convoluted tube which is called ejaculatory duct arises from the seminal vesicle which extends forward and enters into a muscular or copulatory organ which is called penis, or here it is called as cirrus. So this opens into the genital atrium by the male genital aperture, and this genital atrium opens outside through a common gonopore. Okay. And here there are prostate glands also which supports this opening, so which helps in the ejaculation. So ejaculatory duct, they surround this duct. And a bag-like serous sac encloses the seminal vesicle, serous and posterior gland. So this is how the male reproductive structure looks like. So I think there will be figure. Yeah. So this is once I complete the female reproductive system also, we'll see once. So female reproductive system consists of a single ovary, oviduct, uterus, and associated vital area and Mehlis glands. So ovary is a branched tubular structure which is unpaired. It is present on the right side of the body, okay, in front of the testis. So here both male and female reproductive organs are present in the same organism, okay, and it is present behind the ventral sucker. And the walls consist of germinal epithelium, and it produces and releases ova, that is egg, into the cavity. And all branches of the ovary, they unite posteriorly to form a short narrow tube which is called as oviduct. So it runs backward towards the middle line of the body and opens into the vitelline duct. And from the junction of the oviduct and vitelline duct, a long tube which is called uterus is it arises, and it extends up to the genital atrium and opens into the female genital aperture. And this aperture is close to the male genital aperture which is on the left side. And this uterus, it stores the capsules which contains fertilized eggs, okay, and at the end they eject these eggs, so that is known as metraterm, okay. So during breeding period, a short slender tube arises from this terminal and which is called as Laurer's canal, and it runs ventrically upward to open up in the dorsal surface of the body, and this actually serves as a vagina during copulation. So here you can see this is the reproductive system of this organ, which looks very complicated, of course. So all are like they look like branches, okay, small branches, but you have so many organs here. So associated with the female reproductive system, there are two important glands: vitelline glands and vitellaria or Meheli's glands. So vitelline glands are very minute. They are small spherical bodies which are scattered all over the body, and they have fine ducticles which arises from these glands, and they are present on the posterior end of the ovary, okay, and each longitudinal duct present has a anterior and posterior limbs, and they enlarge and they act as a yolk reservoir, okay, and because the yolk has to be present, so it will serve as a nutrition for the larva or egg until it starts its own feeding, okay. And then this oviduct finally forms to the uterus, and these vitelline glands produce yolk cells and consists of yolk which nourishes the embryo and also droplets of the shell forming material, okay. Then Mehlis glands are numerous, that is the number of glands are present, and they surround this oviduct. They open at the junction of the oviduct, and their secretion helps the lubrication of the uterus for the smooth passage of the eggs, okay, and sperms. And here it also helps in the hardening of the eggshell, okay. So this is about the reproductive system of the this worm. Then now finally, so this is about the organization of the body. Now we will discuss about the life cycle. So this liver fluke has a complex life cycle. So it has several larval stages. So it has two digenetic, means two hosts. So first is copulational fertilization. So self-fertilization is rare, okay, in the fasciola hepatica. So although both the organs are present in the same organism, it usually cross-fertilizes through copulation. So the cirrus or penis here of one fluke penetrates into the Lawrence canal of the another fluke and injects spermatozoa or sperms. So the spermatozoa moves to the distal end of the oviduct where already eggs are present from ovary and fertilization occurs internally. Now these fertilized eggs, they receive yolk cells from vitelline glands and the gland gets enclosed by a shell which is made up of chitin. Now you have even the yolk cells inside. So this yolk, it nourishes the embryo. Okay. So the shell hardens and becomes brownish yellow color structure and it has an operculum at one end. And the shell eggs are called as capsules which pass into the uterus where the development actually starts. Okay. These capsules come out of the uterus through gonopore, okay, into the bile duct. So whenever it infects the sheep, bile duct of the sheep, so through gonopore, these capsules come out and they reach the intestine, okay, where after this development, they pass out through the feces. Now each fluke may produce about 5 lakh capsules and a single infected sheep may release 100 million eggs, okay. So therefore, the infection capacity is more here. Okay. So the contamination is very high here. Okay. Yeah, sorry. So here this is the cleavage and larval development. So this adult fluke in the sheep, when it gets infected, it releases the eggs. So first the eggs, they become, so even the larval stage has many stages. Miracidium stage larva, which is free living in the pond water. So then it enters the snail, which is the intermediate host. So where it forms sporocyte, so like it forms a spore-like structure, okay, like a cyst form. Then primary radiaria larva. So it is another stage. Then secondary radia larva, then Cercaria larva. So like this, it transforms from one form of the larva to another stage. Then in the Cercaria larva, it comes out of the snail and again it enters into the water. So it forms an encystment. So it forms a cyst and it stays there in the water plants. Meta-Cercaria stage only gets encysted, okay. Then this, when a sheep it eats that plant, so again that enters into the sheep. So again it continues. So it like that the life cycle continues. So the cleavage starts while eggs are still in the uterus, and at first the zygote divides into two unequal cells, which are propagatory cells, which are small, and somatic cells, which are large. And somatic cells, they divide and form the ectoderm of the larva. Later, the propagatory cells divide into two cells again, which form endoderm and mesoderm. So now you have three germ layers: ectoderm, endoderm, and mesoderm. Then the caps, they form the capsule and they come in contact with the damp areas. And the development usually takes place at a temperature, optimum temperature of 22 to 25 degrees centigrade. Okay, as I told you, there are different larval stages. So we'll briefly discuss about them. So Miracidium larva. So this under favorable conditions, first the shell differentiated into ciliated Miracidium larva within 4 to 15 days, and it produces a proteolytic enzyme which digests the protein. which dissolves the cementing material of operculum and emerges out into the water. And this larva, which is ciliated, which is at broad at the anterior end, which is non-ciliated, and it has head-like lobe. Okay, and the body is covered with the plates, and number of arrangements of cells are present. And beneath this epidermal layers, there is subepidermal musculature. So muscles are present, and it has outer circular and inner longitudinal fibers. Okay, and with it has a cavity, internal cavity. In the cavity, it has apical papilla and granular sac. Okay, which is called apical gland. And you have another penetration gland also. Okay, and the excretion is through flame cells. And in the posterior end, you have germ cells, which are called germ balls, which are helpful in the developing embryos. This meracidium do not feed, and it swims actively in the water, and it enters the snail, which is limnia, and which is the intermediate host for the liver fluke. And if fails to reach the snail, usually after, like within 24 hours, it dies. But within 24 hours, if it can find the snail, it will enter the snail. So when it comes in contact with the snail, it adheres the snail body through papilla, and then through penetration glands it enters. Okay, it remains there inside the snail, and it forms a sporocyst, and again it forms different forms of larva, which is called as radia larva. Okay, this sporocyst consists of germ balls. Okay, and which is made up of muscles again, and its germ balls develop into another type of larva, which is called radia larva, which was named after the scientist Reddy. And each sporocyst produces 5 to 8 radia. And this radia larva, again, it has two stages. So it has small anterior mouth and a muscular collar surrounded by muscular collar, and a birth port lies close and behind the collar. And then you have a mouth there. It leads to the small muzzle. Okay, then an intestine sac-like structure, pharyngeal glands, then protonephridia is present, which is the excretory system, excretory duct. Then you have germ balls and mesenchyma. So this is the active larva. It goes through the host tissue with a muscular contraction, and then where it grows and produces the other stage, second generation also will be produced, and the next larval stage is Cercaria larva, and each radia larva produces 14 to 20 Cercaria, which leaves the body through birth pore into the digestive gland of the host. This Cercaria larva, it resembles the adult fluke, and the body wall is present, and the spinules are present, and they finally enters the next larval stage, which is called as metacercaria, which have anterior and ventral suckers, which are mostly resembles as adult flukes. They have digestive, excretory, and genital system, which are similar to the adult flukes. Now, these metacercaria, they get released and they will get encysted. That is, encystment takes place. So they will form a cyst. So
Madam, what questions DJ now, please?
yeah, I told. Let me complete the topic. In between, how can I? So this is a single topic. You cannot get. Okay, so let me complete this. Then I'll discuss. Okay. So the metacercaria has round body and it has hard cyst. So it will form a cyst, and it has excretory bladder and opens into the single pore. Okay. Now this enters into the the adult sheep. Okay, then it completes its life history. So it has different larval stages. So all the larval stages I have discussed. Meracidium, Sporosis, Radia, Cercaria, all these are formed from the germ cells. Okay, ectoderm, endoderm, these are the germ cells. Then, which are called as, okay, they are called germ layers. So from the germ layers, you have germ cells, which are called propagatory cells. So from propagatory cells only, all these are appears. So then, when the metacercaria is followed by the sheep or goat through vegetation, it enters the sheep. And the cyst wall, which is present, that is digested by the gastric juice in the intestinal wall of the cavity, and it penetrates into the liver, where it lives at 5 to 6 weeks, and there it starts causing the liver rot. It damages the liver, or fasciolosis is present. Again, it enters the bile duct, where it remains there. After entering into the host body, it will remain there for 11 to 13 weeks to start laying the eggs. And then it completes the life history. And the pathogenicity. So it mainly infects the liver. It is due to the irritation which is caused by the cuticular spines. So all the spines, they cause the irritation in the liver. And during migration, they cause the liver damage, liver tissue damage, and even the bile duct also. So they cause jaundice, obstructive jaundice, and they produce cystic dilatation of the bile ducts. Okay, and gallstones also will be followed. So therefore, it results in the pain, swelling of the abdomen, loss of weight, eye sockets become pale, and enlarged liver. So all these will be formed, and finally, if not treated, the animal dies. So the control measures are, so mainly treatment is available by hexachloromethane and tetrachloroethane, which can destroy these flukes. But preventive measures is by killing the intermediate host by using sprinkling the copper sulfate solution on the ponds, okay, where the rearing is more, like rearing of the cattle. And the snails also can be killed, okay, by drying the regions for the larger periods of time. So this is about this fasciola. Okay, so yeah, so tinea solium is also important. Okay, I will just discuss, I will just let you know some important topics. Let us see how much we can complete. So whatever the topics I have discussed so far, so in the animal kingdom, so in the protozoa, so these are important topics because you go through the type studies mainly important. Okay, so leishmania is important. So the type studies, malaria is also important. Malarial parasite and trypanosoma. So these three and fasciola. Now whatever I told today, then tinea solium. So these five are important topics, like type studies. Porifera, I don't think that is much more important. But compared to porifera, you more focus on this thing, the first topic.
Ma'am, repeat once again.
Sorry,
Repeat once again. Fasciola, malaria, parasite, and
first three topics in the. There are only three topics there, right? So here you have three topics, right? In the protozoa, you have three topics, right? Three case studies. You have only three case studies, three diseases, right? Leishmaniasis and Entamoeba histolytica. Yeah, four you have. Then malarial parasite, trypanosoma. Okay. So see, case studies are important because you cannot expect which case study you may get as an essay answer, right? So you
Yes.
go through these four and that too, fasciola hepatica and trypanosoma. So yeah, what is that tapeworm? Okay. So unit 2 is important. Unit 3. So other than the case studies, so you can just have the general characteristics classification of other or animals. Okay. Are you getting my point? For every unit. So for every topic, you prepare the general classification and um, what you call,
Character.
yeah, general characteristics. You take one or two examples of each one. Okay. And organization in brief that you prepare for all the topics, because that can come for small questions, short answers. Okay.
Okay.
But for essay answers, you can expect type studies. So that type studies, you have that four, and here you have two. So today whatever I have discussed. Platyhelminths, you have two, right? Two type studies. So these two also important. So I cannot say which type study you will get. Okay. So, but if you study one, the other will also be easy only, because the general characteristics all will remain same for both if it belongs to the same phylum, right?
Yes.
So other than this, the next topic is so Ascalemanthus is not much, means when compared to Platyhelminthes, this you can just that is more important, right? Then Annelida is important. Yeah, Annelida is important. Okay. So you have a type study, right? Yeah, by for today you take these important first topics. Again, I will go through once. So in the Annelida and Arthropoda, if I find any important topics, I will discuss in the next class. So next class is the last class, right? Next Sunday.
Yes.
So finally, I will tell you on that day.
Ma'am, in last class, just tell us full chapters important questions, ma'am. Previous chapter
Sorry.
we don't have important questions. For previous chapters we don't have important questions.
Previous chapter, which chapter? Name the chapter.
Hello.
Hello? Yeah, please tell me the names of the chapters. Previous chapters means which? Hello? Please tell me the
Protozoa,
names
ma'am.
of.
Protozoa,
Sorry,
porifera. Protozoa 1, block 1, ma'am.
that's what I told, no?
You told, ma'am.
Just now I told, no?
That is only previous chapter she asked.
Yeah, that's what I told, no, already. So important question, I told
For two
Desh.
full units, ma'am.
Sorry. Not taking interest. You just tell me clearly, like, are you there? Sorry, what do you want? Tell me clearly so that I can tell you. I'm not understanding. Like,
Maybe she is mute, ma'am.
please unmute your mic and speak.
Ma'am, you are preparing question paper.
I won't prepare. I'm just resource person. I will just. That's why I'm telling. I cannot expect which questions comes from which topics. I can tell only important topics to focus.
Yes, ma'am.
Don't think that. Don't say again. She madam told this topic. This topic didn't come in the exam. No. If you are in that mood, I cannot discuss any important nothing. You have to study the complete textbook.
Okay. Just ask.
Okay.
Okay.
No, I'm just telling. Like there is no important actually because you cannot expect like all are important. No, all are important. That's why they have given in the syllabus. Only for
Yes.
the time being, you can just focus on more important ones
Topics.
which
Yes.
which have more importance economically and all that, which you can expect, but it is not guaranteed.
But in that arthropoda, ma'am, that prawn
Sorry, arthropoda.
Paleman, that prawn system, it was there, ma'am, in practicals, like
Yeah, that's
appendages and all.
what I'm telling. See, ma, anything can come. So that you cannot say arthropoda will be there, annelida will be there. From every topic, you will have one topic in the practicals, one topic in the. It is not like it is not like a school. No, you can expect the same question. You
Yeah,
cannot. You can directly exclude this. No, this is not school, right?
yeah, right.
You have to study everything. Now you are going into professional thing. You are a
No,
zoologist, right?
actually, it is so vast, so that's why.
That, that's right. But you have to study. There is no other option.
Okay, ma'am.
Just for passing means you can just focus. But you want to take it as a career. Now after BSc you will do MSc. You cannot just I'll read only NLED, I will go to MSc means no, it you have to knowledge of all the topics, right?
Yes, ma'am.
Only for the passing, for the time being, you can just have okay, you can read one topic which is more important and you can exclude. But it is good that if you study all,
All
if you
yes.
have time, you study all the topics.
Okay.
I will give you the tips like for any topic. So all these are the classification. All these are different phylums, right? So
Yes,
for
ma'am.
all phylums, what you do is just prepare
General
like
topic.
a table or something. Don't read each and everything. Don't like a text. Don't remember like a text. You
Yes.
cannot remember. That is impossible. But mostly all the organisms will have organ systems. Whether it is having mouth or digestive system, respiratory system, excretory system, nervous system, like this, that will be
Yes.
common. Okay, what is a reproductive system? Excretory system. Whether it is present, yeah, present. If it is present, what organs? If excretory system is present, what? So here today we discussed flame cells are used as a excretory organs. If it is a digestive system, so what are present? Mouth, pharynx, esophagus, intestines, and anus is absent. Like that, you write down
Okay.
2 or 3 times. If you read, you will remember. Okay, even after some years also, you just go through the table, it will be easy for you. So just prepare a small introduction about each topic, just 3-4 lines. Introduction, then general characteristics, classification. So what are the classes, or like that you prepare for all the topics.
Okay, ma'am.
In 2 to 3 days, you can prepare, right, for all the topics. Then each topic, you go through what are the type studies. So what are the examples they have given. So now in the first one, you have given, you have 3 type studies here, right? So you can prepare any 2 type studies. So if all belongs to same phylum, some points will be definitely common.
Common, yes.
Okay, so their organization will be common. Okay, body organization. If you
Yes, ma'am.
remember few points, you can write on your own. It's okay if you forget some points also. There is no harm. But some points you can remember, no? So
Yes,
if you have any figures, you focus on the figures mainly. Then disease, mainly the life cycle is important in, because that differs from each type study to other type study, right? You
yes,
remember that life cycle. So like that, if you prepare, no, it won't take much time, and even you can remember easily. So first you focus on most important topics. Then if still you have time, you can focus on the other topics also,
okay, ma'am.
right? So even annelids are more important. I told you. Even in the arthropod, now you are telling that it is there in your prawns,
Practically.
is there in your practicals. So during that time when you're writing records, you might have covered some points, right?
Yes, ma'am.
Already you might have written no records.
Records written, ma'am,
Yes, yes.
and they have given nervous system of
Yeah,
prawn.
nervous system. So already nervous system, you might have got some points.
Yes, ma'am.
You focus on the other points which are not covered. So one topic will be covered, right?
Yes,
So like that, you both mix. The practicals is not separate and theory is not separate. Theory, what you are studying, you are applying in the practicals. That is.
yes.
Now, whatever you are studying, you are lively seeing that in your
So
practice.
that manual also we can refer, ma'am. The
Yeah, definitely,
practical
no
for
problem.
writing.
Yes, if you remember that points, if you feel that is more easy,
Yes,
you can
that,
write that points. It will be same only, right?
yes, ma'am.
Whichever, if this textbook is difficult, no, you can also refer internet. If you
Yes,
find
ma'am.
some points simple, any
Okay.
source is okay, but you try to focus on the topics. You make it simple. You first understand. You make it simple. If you understand, you can write it on your own also. Nothing to write as it is. You have to write me. Then nothing like that.
Okay.
Okay. So
Okay,
don't
ma'am.
worry much about important topics. So only topics will come from either your theory or your practicals. You keep both of them. First you prepare you prepare your own notes. Then once you prepare your own notes, you will get to know. Like one reading will be over at that time.
Yes, ma'am.
Just refer that table or that notes when you are going for the exam. So that will be very easy. Okay.
Okay, ma'am.
Okay, fine. Again, in the next topic, in next class, if I find any other very important topics, I will discuss.
Okay, ma'am. Thank
Okay,
you.
fine. Yeah, thank you.
Thank you.
Yeah, yeah. Thank you.
Thank you.